No worries. All right, chapter 13, hemoglobinopathies. Really important chapter. There will be several questions probably on your boards about hemoglobinopathies because they're one of the most important things that you need to know about hematology. Okay, hemoglobinopathies. So what are they? So basically, it's genetically determined abnormalities of the structure or synthesis of the hemoglobin molecule. In this case, we're not talking about heme in general, but we're talking about the whole molecule, hemoglobin in this case. So most of the time, though, you listen to me close, most of the time, heme is normal. It is the globin chain that is abnormal. Okay? The globin chain is abnormal in these hemoglobinopathies. Okay? So there's two forms of hemoglobinopathies. You can have qualitative forms and you can have quantitative forms. Okay? Qualitative is structurally abnormal. Okay? Quantitative, of course, is quantity. There's not enough globin chains to make up normal hemoglobin. Okay? So qualitative is usually what we consider the hemoglobinopathies. Okay? So things like sickle cell anemia, hemoglobin SC, hemoglobin C disease, these all are hemoglobinopathies and they're usually qualitative. Okay? It has to do with the structure of the globin molecules. Whereas quantitative, they're called thalassemias. Okay? Thalassemias are missing globin chains. We can't make enough globin chains to form hemoglobin normally. Okay? So, where do these genetic mutations occur? It's usually in the coding regions. So you remember we've got two different things. Go back to biology, and you have a gene. Okay? And that gene has parts to it. Okay? And in the gene, you have what's called introns and exons. Okay? Now, these exons here are where the actual code is. And a lot of times, that's the only thing that gets coded for the actual protein that's being made. Okay? So when we look at this, there's usually an abnormality. And most of the time, as we look through, they are simply what we call point mutations. Okay? So, as we're making our messenger RNA, like we talked about not too long ago, we're making our messenger RNA. So, we're going up A, T, C, G, C, C, A, T. Okay? So what happens when we're making our messenger RNA, instead of having a U here, an A gets put in. Okay? Now I know we've got that little checker that's supposed to go up there through there and check it, but sometimes it gets missed. Okay? So instead of this being AUG, it ends up being AAG. Okay? So this, remember, is a codon. Remember, talked about three nucleic acids are considered a codon. Okay? And what do those codons do? What do codons do? They code for what? Huh? No, they code for proteins, but what do three nucleic acid codons do? What is their main job? Transfer. To summon what? Huh? What was that guy? Transfer RNA, right? Remember, this is what the amino acid is. This is the code for the amino acid. This one has an anticodon. In this case, it'd be UUC, right? And it's going to call that particular amino acid into place, all right? But because we know it was supposed to have been that, this right here gets called, so it brings in and puts in one wrong amino acid, okay? And we're going to talk about it. And 9 times out of 10, every one of these variant hemoglobins that we're going to talk about, that's the problem. Is one substitution of an amino acid causes it to be totally different than just hemoglobin A, okay? Yeah.
What's intron?
Huh?
What's in?
They're just like excess nucleic acids. They don't really have any code or anything in them, okay? All right, so genetic mutations in the coding region of the globin gene. Okay, so where are the globin genes at? What 2 chromosomes got our globin genes? 16 and 11, right? Okay, so results in amino acid deletions or substitutions in that globin chain, which therefore throws off our protein. What do you mean?
What does it cause if missing that one?
If it comes out AAG.
So if it's missing the 1, it just makes a whole new protein.
So
Does that make sense? So it doesn't do what it's normally supposed to do. Okay, so normally we know that hemoglobin A, right? A1, hemoglobin A, the adult hemoglobin that we said makes up what? 95 to 97% of the hemoglobin in an adult, right? Mm-hmm. We know it's made up of alpha 2 and beta 2, right? Alright? Now, most of the time, this is not our problem. The alpha gene, why is it never going to be a problem for us?
everything?
Huh?
It's in all the other.
Well, it is in everything, but why is it going to be not going to be a problem? That a girl, because remember it's on the 16th chromosome, and it's an alpha 1, alpha 2, alpha 1, alpha 2, and these two are identical to each other, right? So we got 4 chances of making this one right, okay? Now what about this guy though? He's over there on the 11th one, right? And how many copies of him? He just got 2. So if one of them gets screwed up, he's going to be our problem. And that's usually where it's at, okay? Usually this is the problem, all right? So even though it's 2 alpha, 2 beta, but because this was normal, this is abnormal, this one gets inserted. So instead of us having amino acid here, 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 and let's say, and I'm going to tell you the real thing, okay? Right here in this position, it should be glutamic acid. Okay? That's normal. That's what it should be. But because this got mutated to this, now we have valine. Valine gets placed in instead of glutamic acid. This is what gives us the big one that we're going to talk most about, hemoglobin S, which is sickle cell. Okay? All right? Okay, questions about that? Does that kind of make sense a little bit, what we're talking about when we have a mutation? All right, so let's move on to the next section then, okay? So, causes structural variation in one globin chain, and the most common clinical disorder that you're going to see is sickle cell anemia, okay? Sickle cell anemia is known as hemoglobin S disease, okay? All right. So, like we talked about a while ago, here's the slide for it. I always get ahead of myself, but it doesn't matter. Quantitative defects, when we talk about the number, okay? There's actually 2 types of thalassemia. I'm going to go ahead and introduce this, even though there's a whole nother chapter. Chapter 14 is all about thalassemias. There are 2 types. There are alpha thalassemias. And then there are beta thalassemias. Okay? Alpha ones tend to be more severe. Okay? And the reason they end up being more severe is because they're necessary for every adult hemoglobin that we have, right? Alpha, 2 alpha, 2 beta gives us hemoglobin A. 2 alpha, 2 gamma give us hemoglobin F. 2 alpha, 2 delta gives us hemoglobin A2, right? So without alpha, if there's a problem with these 4 alpha genes, either just one of them, even if just one of them's messed up, it still hurts our hemoglobin production, okay? Now, luckily for us, our body can compensate for it, okay, because we've got those 3 other genes, and we see a really, really mild anemia in these patients. We still see anemias, okay? But there's actually one that all 4 are deleted, okay? And this is actually called, Dropsis fatalis. Sounds like Harry Potter. It's called Hydropsis fatalis. Baby never survives. They can't survive. Because if they have no alpha genes, what kind of hemoglobin do they make their whole life?
Ridiculous.
Huh?
Ridiculous.
No. What kind of hemoglobin do they make? Y'all remember?
Oh, yeah.
And if I remember right, it's Gower 1 and Portland that have the 2 zeta genes. Okay? Because there's nothing wrong with their zeta genes. Their zeta genes will form. So what happens is a lot of times they will survive up until a certain age of gestation. Until normally, remember, this gene gets kicked in and they start making fetal hemoglobin in about, like 18 or 20 weeks gestation. And then after that, it all is hemoglobin F, right? Which is 2 alpha and 2 gamma. But because those genes are deleted, a baby is usually born stillbirth. They don't survive. And it's called Hydropsis fatalis. It doesn't matter. We're going to talk about it later on, but just to introduce it to you, okay? So thalassemias can be very detrimental, especially the alpha ones. You could also have combination effects. There are people who have hemoglobin S but also have thalassemia, okay? Usually it is beta thalassemia, okay? But they have a form of thalassemia plus a hemoglobinopathy qualitative form, okay? So that's one of the things that we'll talk about a little later on, is you can actually have combinations of defects, okay, as you go. And we're going to talk about one that's called hemoglobin SC disease. They actually produce hemoglobin S and they also produce hemoglobin C, okay? So that would be a combination of defects in the patient, okay? All right. So hemoglobinopathies, of course, can be associated with chronic anemias, which usually they are. Usually they don't have any problem acutely unless they go into what's called a crisis. Some of the hemoglobinopathies, the abnormal hemoglobins, actually function fine. So your patient is actually asymptomatic. They don't have any problems. You never know they have a hemoglobinopathy unless you did a hemoglobin electrophoresis on them. Okay? So what are these that we're going to talk about? We're going to talk about alpha, beta, delta, and gamma mutations as we go. The severity of the hemolysis depends on which one of these it is. Okay? And then we also always have compensatory production of other globin change. Okay? So, for instance, when we talk about beta thalassemia a little later on, okay? In beta thalassemia, we make alpha just like we're supposed to, but there's an abnormality in the beta chains. Either they're both deleted or just one of them is deleted. Okay? So instead of dying, our body just upregulates the gene production of gamma, okay, and A2. So you see an increase in gamma and delta, so you end up seeing an increase in hemoglobin F and hemoglobin A2 in patients that have beta thalassemia, okay? Now, alpha thalassemias, it does the same thing, but we'll talk about it. It's a little weirder than the beta thalassemias, okay? All right? So sometimes it can happen the same way in like hemoglobin S, hemoglobin C. They will actually upregulate their fetal hemoglobin a little bit to help compensate, okay? All right, and we'll talk why that's a problem. We've talked about it already, but I'll remind you guys, okay? So worldwide distribution of hemoglobinopathies. Again, this is not a very Caucasian thing. There are very, very few Caucasians that have hemoglobinopathies. African Americans are one of the main groups. The reason is because hemoglobin S is very, very prominent. Okay, ethnic groups from the Mediterranean basin, so like Iraq, Iran, and these areas. Okay, those people, the Middle Eastern cultures, they can tend to have some hemoglobinopathies, okay? And then Southeast Asia. When we talk about Southeast Asia, we're talking about places like Vietnam, Laos, Cambodia, those kind of places, okay? India even, all right? Those places we see lots of increases in hemoglobinopathies, and we'll talk more about those. I think it's hemoglobin E is very prominent in Southeast Asia, but sometimes, and y'all don't look at me funny, there is a rare chance that people who have Southeast Asian ethnic background can actually develop a form of sickle cell, okay? Hemoglobin S disease, even though we always think about African Americans being sickle cell, okay? Later on, we're going to talk about this. Having sickle cell is actually an advantage for some Africans who live in Africa, okay? African blacks, and we'll talk about that in just a minute, okay? It's believed to be the most common lethal hereditary diseases in humans, hemoglobinopathies. And the reason it's lethal is because if you can't carry hemoglobin and oxygen like you need to, then you die, right? We have to have oxygen to be able to live, okay? So, what are the most common hemoglobinopathies? Usually they're inherited structural changes in one of the globin chains, which is what we talked about. And the majority of them are the beta, which is why they're a problem for us, because that makes up 95 to 97% of our hemoglobin, okay? So synthesis of the abnormal chains usually not significantly impaired. Any of the globin chains can be affected. We talked about that, but the most common one is sickle cell anemia, okay? Sickle cell anemia, all right? There's over 1,100 abnormal hemoglobins in the world that we know about today. Every one of these are due to genetics and phenotypic expression. So what's the difference between genetics and phenotypic? What does genotype mean? That means your genes. What's your phenotype? So what you express on that gene, right? So that's the reason some of these things can be genetic, and that's where the problem is, but sometimes it's phenotypic also. And most of the time, like we talked about earlier, sometimes, rarely, these patients can have a genetic abnormality, but their phenotypic is fine because they're asymptomatic to it. In other words, it does what it's supposed to do, and they have no anemia from it, okay? But a lot of these, it's genetic abnormality, but it also produces an abnormal phenotype, okay? For instance, sickle cell, okay? Hemoglobin S is definitely a phenotypic thing, okay? Most mutations are seen in the beta chain, but there are a few rare ones that do affect hemoglobin F. So this is expressed during fetal development, of course. Variants of hemoglobin F unlikely to be detectable after 3 to 6 months of age, okay? Because most people are not doing any kind of test on babies. And by the time they reach 3 to 6 months, what kind of hemoglobin's kicked in? Hemoglobin A. So we're not even worried about that gamma chain at that point because naturally our body downregulates gamma production. And so it doesn't cause us a problem. Okay, or at least we don't ever pick it up. Okay, all right. So this is basically the number of variants in the alpha, number of variants in the beta, the delta, the gamma A, and the gamma G. Okay, so very few of these, and one reason there's probably very few of these is just because we don't search for them, we don't look for them that often. Okay, there may actually be more than that. But because we don't test for it, there's not really anything that is done. Okay? All right, so questions about that. So by far, beta is the one that has the most mutations. Okay? All right, hemoglobin A2, we know is a minor hemoglobin, unlikely to cause any clinical complications because we make so little of it. Even if it was abnormal, it wouldn't really matter. Remember, hemoglobin A2 is 2 alpha and 2 delta genes, or globin chains, okay? 2 alpha and 2 delta. All right? So, let's talk about the alpha gene in general, okay, for just a few minutes, okay? It is duplicated on each of the 16 chromosomes. We've talked about that. So, mutation in a single gene doesn't really matter. We don't really care about it that much because it's only going to produce small amounts of an abnormal hemoglobin. So usually the alpha is not going to cause us any problems, okay? So less likely to cause any clinical complications. All right, now the beta gene though, on the other hand, is a problem because we only have a single copy on each chromosome. So we have two copies in our human body. This one is associated with most clinical phenotypes of a problem. Okay, if the mutation occurs, we usually do have phenotypic problems with this. Okay, for instance, hemoglobin S, the cells will sickle. Hemoglobin C forms a crystal. We'll talk about it in just a little bit. And hemoglobin SC in combination also forms a crystal plus sickle cells. Okay. So it causes us lots of problems. And we know that beta is the common globin chain of hemoglobin A. All right. Single amino acid substitutions or deletions is usually our problem. The clinical symptoms vary. It just depends. They can go from no abnormalities and no problems to basically altered function of the hemoglobin. Okay. When we talk about function, we know hemoglobin's main job is to carry oxygen. So one of the big things we worry about is oxygen affinity, right? We need it to have that attraction, but we also need the avidity to be low to let it go, right? So when we get into like talking about hemoglobin F compensating, that's a problem because hemoglobin F actually has a really, really high affinity for oxygen, but it also has a high avidity for it. And so even though we might try to compensate, a lot of times we don't do the job well because hemoglobin F doesn't want to give up the oxygen molecule good enough. Okay? Stability. This is what our problem is with insolubility. These are the problems with like hemoglobin S and hemoglobin SC and hemoglobin C diseases. Okay? All right? Questions so far, guys, on this? All right, so let's talk about how we detect hemoglobinopathies. And this is going to be really, really important for you guys to know. All right, so there are screening tests that we do. And we're going to talk a little bit about those later on. But the one that we want to talk about mostly is electrophoresis. Okay? So I kind of talked a little bit about electrophoresis the other day in class when we were talking about serum protein electrophoresis. Now this is a protein electrophoresis, but it's a specialized protein electrophoresis. Because most of the time we're looking at serum, okay? So we talked about how that we have a chamber, okay, and then we put a fluid in it, some kind of an electrolyte, okay, some kind of medium, okay, and then we have like a gel agar in it. They can be gel, okay, so they can be agarose gel, okay, or they can be acrylamide, I think that's how it's spelled, okay. Both of these are two types of medium that can be used, okay? And it all depends on what your pH is, okay? So let's think about how we talked about amino acids make proteins, right? Okay, so if you remember when we talked about proteins in hemoglobin, and a little bit in the first part of this, and this is all basic biology, okay? We know that amino acids can have charges on them, right? They can have net charges, a positive charge, and negative charges, right? Remember us talking about that? Okay? So, my question is this how our proteins stay? No, this is that primary form, right? But then we go ahead and we form that secondary form where it does this and starts bending a little bit, right? And then we get the tertiary form like this, okay? This is what causes us to have properties of chemical properties, okay? Or electrical properties. The reason it does that is because if this one binds over close to this one, and we've got more negatives here, then our charge becomes negative. Does that make sense? Okay, because this is negative, this is negative. Let's say this one's negative, this one, this one, this one, and this one are all negative. Okay? So when this thing gets all bound up close to each other, and those charges get close to each other, you end up getting a buildup of those charges. Does that make sense? So in other words, by the time we get done, our protein has a negative charge on it. Okay? So if it has a negative charge, which end is it going to be attracted to? It's going to be attracted to this end, right? Okay? So if we know that our protein has an overall negative charge, we want to set it on this end of our gel, or our medium, whichever one it is. Does that make sense? Because we want it to go that direction in our flow. Okay? So you have a couple of different types of medium that you can use, and basically whichever one you're using, and the pH. Okay? Now pH changes things, right? So remember, well, I don't know if I told you guys this. 3 things affect proteins, okay? Heat, pH, and radiation, okay? They change proteins, all right? So if I put you in a tanning bed, right? Correct? And I leave you in there and leave it on high for multiple days, you're going to burn, right? But eventually, what are you also going to develop? Cancer. Why are you developing cancer? The exposure to the light, right? What kind of light is that? UV light, which is also called, starts with an R, radiation, right? It's a form of radiation. So the reason you develop cancer is because you've actually mutated your proteins, and those cells lose their brain. And then they start multiplying, okay? Does that make sense? Okay? So when I was teaching high school, I would use an egg. I would take an egg, and I would put it out in a pan, cold pan, and we would talk about the properties of the albumin. We'd talk about how the white is albumin, which is a protein, all right? Then what would I do? I cut the heat on. And all of a sudden, the heat started changing that clear albumin to white albumin. Is it still albumin? Yes, it is. All I did is I went from this structure to that structure. I just changed it. I denatured it. I moved it around. Okay? Right? Okay? Then I would take an egg and put it in a pan, and then I would pour like 12 molar hydrochloric acid on top of it. Guess what happened to the whites? It changed again. Why? Because I went from this, now it was more like that. Because I denatured it in a different way, based off of pH. Okay? So what I'm getting at is, okay, if I change the pH of the surrounding molecules, okay, where all of these were negative because they were really close to another negative, now they may be positive because they've gotten close to more positive amino acids. Okay? Does that make sense to you? All right? Because when you, well, let's think about it. So let's say that I've got, here, let's do something simple. Okay? So let's say that I've got something that's negative, negative, positive, positive, negative, 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 positive. Okay? So if I got two pieces and I push them together, what do I have more charges of?
Negative.
I have more negative charges, right? Okay? So, because I pushed them that close together, all right? Now, let me tell you this, okay? These had negative charges on them, right? And these also had positive charges on them, okay? But because these two were closer together, it gave off an overall negative charge because there was more energy holding them together in the negative form. Okay? Does that make sense? All right? So now let's say now I come in and straighten this strand out. Right? So now I've got negative, positive, 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 negative, 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 positive, positive, positive here. Okay? Now in this case, because I have more positives and they're all bunched in here together, what's my overall charge going to be? Positive. So just because I changed the structure, and how, you got to remember, these are all little chemical bonds in there together, okay? So the more chemical bonds that have charges on them, the more your net charge is going to be, okay? Does that make sense? All right? Do you understand net and what I'm talking about when I say net charge? Okay? All right, do you work now? You have a job? No? Have you ever had a job?
Yeah.
Okay, so you've had a job. So when you got a paycheck,
Got a gross amount in there.
you had this amount. This was what you made, but you only brought home that. That's your net. So this is what your total bring home was. That's what you had. That's what you get to use, right? Okay? So that's what these charges are. When we call it net charge, we may have a bunch of more positives and negatives, but what I end up being able to use at the end is positive. Does that make sense? And what I get to use at the end is negative on this one. Does that make sense? So this, though, can be adjusted or changed by changing pH. Okay? So you're going to see a pH of, I think it's 6.5 and a pH of 8.6. And those numbers might be off just a little bit, but I'm pretty sure that's the right ones. Okay? These are the two most common pHs that we use in electrophoresis. Okay? Notice what they are. One is very acidic and one is very basic or alkaline. Okay? So that makes a difference, right? Because how things act in a base are totally different than how they act in an acid. And it changes their structure based off of that. Okay? So, based off of where the pH is, you may have your origin at this point, okay? Or you may have your origin in the middle, okay? If your origin is in the middle, most of the time you're going to have positive and negative samples, charges. Does that make sense? So, like here, you may get a band that shows up here and here, but you also may get a band that shows up here and here, okay? So what that means is if this was my negative and this is my positive like we have on our thing, these two were negatively charged and these two were positively charged. Does that make sense? Okay? But usually, if they're down here, they're all negative charged right there. And so they're all going to be pulled to that positive end. Does that make sense? That makes sense? All right. So that's the one important thing, okay, is the electrical charge. What's another thing that also affects how they flow? Well, yeah, diffusion would be good, but what affects diffusion? All right. Ken, I'm going to say you weigh $105, if that, okay? Joseph weighs about 235, okay? Is it going to be easier to throw my fat self in the air or Ken? Very good. Same way here. If I want these things to move, they're going to move based off charge, but they're also going to be moving based off of their size, okay? Little tiny ones that have a good charge, they're going to come over here, they're going to show up way down here close to the positive, okay? Big fat ones like me are going to be right here close to the origin. They may have a not positive charge or a negative charge because they did move toward the positive, but they're so large that they don't move as fast. Does that make sense? Okay? So charge and size play a big role in electrophoresis. Very, very important. Okay? Charge and size make a big difference in electrophoresis. Alright? That makes sense. Okay? This is diagnostic. Listen to me. If I ask you a question, how do we diagnose hemoglobinopathies? You're going to tell me hemoglobin electrophoresis. Okay? There are screening tests that we do all the time. We do them in the lab. Okay? They're solubility tests is what they are. They are not diagnostic though. They are screening tests. For me to diagnose hemoglobinopathies, I have to do electrophoresis. Okay? Qualitative. Alright? Now, you can do quantitative hemoglobinopathies, in other words, the thalassemias, okay? They will form electrophoresis also, okay? But to diagnose them, you must do what? That a girl. Genetic testing. Because remember, that's the problem with them, is a deletion a lot of times of those genes. So you have to do a genetic screen or a genetic test on them. Yes?
Are you going to show us how to do them? Like
In the lab? No, I don't have the equipment to do this in the lab.
video or something?
I may show you. I'm not going to show you a video. I'm going to show you examples of it. And we're going to look at them. Okay? All right? Questions about this?
Say it again. How do we diagnose hemoglobinopathy
Hemoglobin electrophoresis. Thalassemias we diagnose using genetic studies. Alright? So that's pretty much what these few slides right here talk about, is just like what I just told you guys. Okay? Alright. Alright, other tests, of course, you can do is CBC. CBC is usually the main test that we do. We look for the anemia, so low hemoglobin, hematocrit, And then we look at RDW and RBC indices. All right, these can help us also distinguish hemoglobinopathies and thalassemias. Okay, and then of course we can do screening tests for hemoglobin variants. All right, and we'll talk more about those in just a little bit. Okay, now quantify hemoglobin A2 and hemoglobin F. There are lots of things we can do. There's one that's called isoelectric focusing. Basically, you're using an electrical impulse to try to pinpoint which ones you're looking for. Higher performance liquid chromatography, which is very similar to electrophoresis. It just uses a liquid medium instead of a solid medium. And then, of course, capillary or traditional electrophoresis. Okay, so this is a very important. I was off a little bit on my pHs. I apologize. I was doing it from my head. So I wasn't off too bad, but I was off a little bit. So let's correct that. So 8.4, and then what's the other one? 6.2. Okay? But still the same concepts, acid and a base. Okay? So in base pH, all right, in base pH, we use cellulose acetate, which is a form of agarose. Okay? Cellulose acetate. Okay? And then we can have a citrate agar gel, can also be used. All right? And notice in the pH of 8.4, your origin starts on the end. Okay? So in this case, in the pH of 8.4, all of your hemoglobin is negatively charged. Okay? So it's all going to go toward the positive electrode, which is known as the A node. Okay? A node. Right? Notice where your origin is in the pH of 6.2. Okay? When you expose the hemoglobins in a low pH or an acid pH, you have positives and negatives. Okay? So therefore, you've got some that migrate toward the negative, so they're positively charged, and then you have some that migrate toward the positive side, so they're negatively charged. Okay? Every time you do one, listen to me. Every time you do an electrophoresis, you have to have a control. Okay? So basically what I'm talking about is what you're going to see. All right, I'm going to always deal with the basic pH, because I always like my origins to start down here. All right, so this is my sample, okay? Here's one thing I haven't told you guys. So if you looked at this thing sideways, okay, what you would see is you would see a whale right there, okay, depending on how deep it is. And what you do is you would pipette your blood into that whale. Okay? Does that make sense? And then you would set your whale or your media down in the liquid. Okay? And then what's going to happen is it's going to flow through. Okay? And then based off of its size, you're going to get an accumulation. Okay? And as it goes on, going to keep going based off its size, you're going to get another one and another one. Okay? In theory, you should only have 3 bands, right? You should have a band of A1, you should have a band of F, and you should have a band of A2. Because remember, that's the 3 hemoglobins that make up our red blood cells, correct? Okay? So, like this band, if it's A1, should be a darker band. Does that make sense? And then a small band, and then a little bit larger band for the A2. Does that make sense? Okay? Now listen, this is going inside the gel. Okay? It's pulling those proteins inside that gel based off of their size. Okay? So, you're going to run a control. Okay? And that control is going to have, all right, now I'm just going to say this, your control is going to have every type of hemoglobin you're looking for, okay? So you're going to have a band that has for A1, F, A2, S, C, and then SC. Or actually, I'll take that back. You won't have the SC because I'll show you in just a minute, okay? So you're going to have every one of these, okay? And then you're going to run your patients, okay? So this would be your control. And then you're going to have patient 1, patient 2, patient 3, and patient 4, okay? So we're going to run theirs, all right? So we're going to say 1 looks like this. We're going to say okay, so that's what your bands are going to look like. All right, so now let's diagnose our patients based off of our bands. Okay, so we're going to say patient 1. Okay, patient 1 has no A1 there. So we know that this patient's what? Abnormal. That's all I need you to know. They're abnormal because they should definitely have a huge band of A1 because we know, because we're smart, A1, F, and A2 make up an adult hemoglobin variation, right? Okay? With the majority of it being A1. So if I don't have any A1, I know I have a problem already. My patient definitely has a hemoglobinopathy. Okay? Now I have to figure out which one it is. Okay? Well, they've got a little bit of F, but they actually have kind of an increase in F. So that's the compensation that's occurring, right? They've got some A2, they have no S, but look at this big band in the C area. So what does that mean? That means my patient has hemoglobin C disease. And we'll talk about it later on, okay? I'm just throwing these out there, okay? All right? Let's look at patient 2. First thing I notice is what? No A1, right? At all. So I know they're abnormal already, okay? Now look, they've got an increase in F, which that tells me that's a problem because now they're compensating for something, okay? A2, and then they have a band in S, but they don't have another band there. So what does that tell me? That tells me this person has hemoglobin S disease, which is sickle cell. Does that make sense? That makes sense, guys? Okay, listen to me. This is important. You've got to be able to recognize a hemoglobin electrophoresis and what's showing up. Okay? They're always going to give you a key, just like this. Okay? They're going to tell you what the lines represent. Okay? Does that make sense? All you got to do is know what you're looking for based off of the disorders that we've talked about. Okay? All right, so let's look at 3 here. Patient 3. All right? Now this is unique because they do have some A1. But do they have a lot of it? No. So that tells me in my brain that they're normal, but not all the way normal. Right? Okay? So let's look. So they've got an increase in F, an increase in A2. Oh, but they've got S. They're making S. So what does that tell me this patient has? This person has hemoglobin S trait. They're a carrier for hemoglobin S. Okay? So in other words, instead of, because here's the deal, guys. All right? That beta gene, have true sickle cell anemia, both genes have to be BS. In other words, they have that mutation I was talking about earlier with the valine takes the place of glutamic acid in the 6th position of the beta chain. Okay? Does that make sense? Alright? So they have to have both genes messed up for them to produce hemoglobin S. There are lots of African Americans and even Southeast Asians that have this. They have one that is messed up, but one that's still producing A normally, like it's supposed to. Does that make sense?
So that would be the trait?
This is the trait, okay? So this goes back to, you remember basic genetics, homozygous and heterozygous. Okay? What does homo mean? Means the same. Hetero means different, right? So in homozygous, this person would be that. So then they're going to form hemoglobin S. Okay? This person is heterozygous. So they have the S, but they also have a copy of the A1. So they're normal. Okay? So this is also a trait. It's also known as a carrier. Okay? Anytime you see the word carrier, your brain needs to think heterozygous. Does that make sense?
So is that something you get from parents?
Yes.
So if you had a African American parent, white parent, you would have the trait.
Most of the time. Yeah.
Not actual sickle.
If that person, the African American parent, it was either a carrier already or had sickle cell, you know.
But most of the time, if they're mixed, they wouldn't have sickle cell.
No, most biracial kids are never going to have sickle cell. They may have the sickle cell trait. Now, even sickle cell trait can cause a little bit of problem, okay? Because they're still producing a little bit of that hemoglobin S. Does that make sense? But usually it's like 15 to 20% versus 80% of the hemoglobin A1. And so, yeah, it may cause them a problem, but there's enough other cells that have it that doesn't. Okay? All right. So I'm gonna throw something out there for you, and I'm not doing it to make you confused. Okay? So we talked about the other day in our red cells, like very first when we started talking about hemoglobin. We said that normally we should have between 95 and 97% of hemoglobin A1. Okay? F, we said, was about 0 to 2%, depending on the individual. Okay? And then A2 is going to be about, what was it, 2 to 5, 1 to 2, I can't remember, 2 to 5 maybe. No, that's too high. I think it's 1 to 2%. Okay? I think that's about right. I think it's about 1 to 2%. Okay? All right, so a normal individual. So that means this person who has beta A1 homozygous. Okay? It means they're just what they normally produce all the time. Okay? Now let's look at this patient. Okay? That we just said was the beta S, sickle cell trait patient. Okay? So what's going to happen is you're going to see a decrease in this to about 80%, and then this guy, then they're going to start producing S at around, 15% or so. Maybe not even that much to make a difference. Okay? Is that going to happen in every single cell? No, it's not. Because because he has this one normal gene, he may have some red cells that are just like normal, a normal individual. Does that make sense? Just because you have the gene, if you're heterozygous or you're a carrier, doesn't mean you're always going to have that particular one in your hemoglobin. Does that make sense? Or in every single red cell. All right? Now, right the opposite, though. When you have the people have this, what are they going to have in their red cells? They're going to have 95% S because that's the majority of the hemoglobin they're going to make. Does that make sense? Because even though they're abnormal, what happened to that gamma and delta gene? Just naturally. It downregulated, right? No, it's fine. Downgraded, downregulated. When they reached a certain age, their body naturally said, hey, we're going to cut it down one way or the other. Okay? Now, as they get older and they start having problems, and hemoglobin S is not doing the job, it can upregulate a little more so that you may see 5% or even 10% of hemoglobin F to help compensate for not having oxygen. Does that make sense? Okay? But this patient, if they're homozygous for it, this is the only hemoglobin, or the majority of the hemoglobin they're going to have in their red cells, no matter what. Whereas that person who's heterozygous here, they've got both A and B. They may have some cells that are normal, some cells that have more S, back and forth. It's just a random thing when you've got one of each one. Okay? They don't all get together and say, okay, we're going to make all these betas, and we're only going to make 5 of you S's. Does that make sense? It just happens the way our body needs, whichever gene gets cut on for that particular need. Does that make sense? Excellent. Okay? All right. We didn't do our last one, did we? Okay. So our last one, we have no A1, so we know we're abnormal. Have a little more F, a little bit more A2, but then we've got S and we've got C. Okay? So this is that combination that I talked about earlier. This is known as actually hemoglobin SC disease. Okay? And we'll talk about those hemoglobins at what time is it?
10, 12,
10, 12. All right, so give me about 15 more minutes and we'll go. All right? Questions about hemoglobin electrophoresis? Very important. Listen, it's very, very important for you to be able to look at a hemoglobin electrophoresis and interpret the results.
Is there a chart somewhere with the results?
Right?
But it
And then I'm
doesn't
going to
what it
give
is.
you some. I'm going to give you some in
Okay.
class
Okay.
that you have to look at. But I'm saying you need to make sure you understand what the lines mean. Okay? And what it corresponds to and those kind of
Well, are we
things.
gonna have a chart that says what the lines mean and what
Yes.
they are?
You'll always have a control.
Okay.
And you'll always have a line that shows you which is which.
Okay, because it
Okay.
can tell us which disease it is.
No, you got to know that. That's what I'm saying. You're going to see the electrophoresis, and it's going to tell you, okay, this is the sickle cell S, hemoglobin S band. Okay? So if you have a person that has a band in that, they have hemoglobin S. No matter what. So then you got to look at it and see, okay, did they have any A? No. So they're only making S. So that means they have hemoglobin S disease, right? But if they're making A1 and S, I know that they're a carrier. So they are hemoglobin S carriers or traits. That's what I need you to know. You need to know what
But are
hemoglobin
those
is
written down,
from.
the disease is what they have in them?
Yes,
Okay.
yes. The
So we-
disease is what determines what they got.
Okay.
Because that's why we call it hemoglobin S disease, or hemoglobin C disease, or SC disease. Okay? As we go. All right? Make sense? Hope so. Okay? Me too. All right. There are tests also that alter the physical properties, or tests for the altered physical properties. So the stability and the solubility that we were talking about a while ago. Solubility test. Basically, you put blood in a certain solution and you look for cloudiness. Okay? So it becomes a solid. A precipitate test is what we do. Okay? And then, of course, there are molecular techniques. You can actually look and find if the gene is abnormal. Okay? This one, of course, we would use mostly on thalassemias, but we could also use it for sickle cell and the rest of them. Okay? All right. Nomenclature. This one's important. Okay? First abnormal hemoglobin discovered was hemoglobin S. Okay? It was actually called hemoglobin S because of crescent-shaped RBCs looked like a sickle. A sickle, of course, is that thing the Grim Reaper uses, what they used to use to chop wheat and harvest wheat and stuff. So the big blade, okay? So sickle cell, they look like sickles, okay? They look like sickles, all right? Hemoglobin S, first one, okay? But then we started getting other things, other ones. We started discovering other abnormal things, okay? So other hemoglobins give successive letters, okay, of the alphabet. But then we started seeing some weird stuff, okay? We started seeing people in Chicago, all right? A group of people in Chicago had hemoglobin S, all right? But it wasn't like the people in New York City, okay? It was different looking. It was a different mutation. It was a different phonetically a different band, okay? So then they started adding cities to it. They would say hemoglobin S Chicago, hemoglobin S New York, or whatever. I'm just making up crap at this point, okay? But they were different. There was no set thing, okay? So then what they started doing is after we started figuring out that there was a problem with the genes that was causing this, we came up with a new system to label these things, okay? All right, so alphabetical system wasn't sufficient because how many do we have now? What did I tell you guys? How many variants are there? Greater than what? 1,100. There are 1,100 different hemoglobin variants in the world that we know about, and that was in 2014. So I don't know how many there are today, okay? Because of our technology advances, we have discovered other variants, okay? How many letters do we have? 26. Ooh, that hurts us, doesn't it? So we know the alphabet system is not good enough. So what they did is they had to come up with a scientific designation or a standardized designation. And so what they did basically is they looked at the gene that was mutated, okay? So this represents hemoglobin S, okay? This is the nomenclature. This is how it looks now, all right? So what it's saying is we know that in the 6th position of the beta chain at the A3 helix, okay? All right, so let me say that. Probably should have talked about this in in chapter 6 when we're talking about hemoglobin, okay? So let's say that this is one beta chain. This is a beta globin chain, okay? Every time there's a curve or a change, it gets a designated letter, okay? Now I'm just making this up. There's a picture in your book that shows you this, okay? Okay? So let's say we're talking about A3. So 1, 2, 3. That position right there, A3, is where we know the mutation occurs. Okay? So in other words, right here is where our AUG got changed to AAU, or whatever it was, I wrote it on the board. Okay? Does that make sense? Okay? So normally, it was supposed to be glutamic acid. Okay? But glutamic acid is replaced by valine. Okay? Does that make sense? So right here, in this position, hemoglobin A1, which is normal hemoglobin, should have glutamic acid there. Okay? But because it got changed to valine, That's what forms hemoglobin S. Does that make sense? Okay? So this is what you need to know. This is the new nomenclature for hemoglobin S. So if I ask you on test, what is the new nomenclature for hemoglobin S? You're going to tell me beta 6, the 6th position of the beta chain in the A3 helix, glutamic acid is replaced by valine.
What was the question again?
I said, if I asked you what the new nomenclature for hemoglobinopathies, hemoglobin S is, you're going to tell me beta 6, alpha, or A3 helix, glutamic acid is replaced by valine. And it needs to look just like this, okay? Every one of them that we're going to talk about have one of these now, okay? We know what it is. The three I want you to be familiar with is S and C are the two big ones that I want you to be familiar with. And we're going to show you, I'll talk to you about C. And there's actually a chart in your book. When we get there. Page 236. Page 236. Gives you hemoglobin S, hemoglobin E, and hemoglobin C. Hemoglobin E is important. We'll talk about it a little later on. But I'm not as concerned with it as I am S and C. Okay? All right? Does that make sense? What it says, what it means. We good? All right. Elijah's shaking his head, so I'm going to say we got it. All right? So, disease or anemia. All right? So, anytime we talk about disease or anemia, we're talking about homozygous. Okay? So, you're going to see this from here on out. You're going to see it written like this. Sorry, I think it's actually, they use a capital letter. Okay? Beta A is normal, right? Because that's what we should make. So if I have a person that has beta A and beta A, okay, and they may put them up here, and that's, I think that's how it actually is. My bad. Okay? I think they're actually superscripts instead of subscripts. Okay? So let me take that off right there. Okay? If I have this person, they are normal. That's a normal individual. So they've got 2 A's. They're producing hemoglobin A1. Okay? This person is what? Heterozygous. So they are a sickle cell trait or sickle cell carrier. Okay? All right? This person is what? They're sickle cell. All right? That's another way of us talking about the same thing that we were talking about over here with the heterozygous, homozygous. Okay?
The BA, BS is the trait.
Correct. Okay? Because it's producing some A1, but it's also producing some S.
BS, BS.
And SS would be sickle cell.
And would it be homo?
Yes. If they're the same, it's homozygous. Okay? Alright, so when we talk about a disease or anemia, we're talking about homozygous. Okay? Homozygous. That means they have both beta genes carry the abnormality. Alright? Trait is the opposite. It's heterozygous. So they have one normal one and one abnormal one. It can be the S, it can be the C, it can be the E, it can be the D, the G, the other 1,000 other things that there are. Right? Okay? But either way, one's going to be normal and one's going to be abnormal on this. Okay? All right? Questions about the introduction? about hemoglobinopathies.
Traits ahead of guys and anemia is
Homozygous. That is correct. All right.